Thank you. Next, it gives me great pleasure to introduce my friend and colleague, Dr. Katie Kelley, who's a GI oncologist at UCSF, who specializes in hepatobiliary and gastrointestinal cancers. She served as the executive officer of the Translational Research Program of the Alliance for Clinical Trials in Oncology and has been a member of the National Comprehensive Cancer Network Hepatobiliary Panel since 2010. Thank you.
Thank you so much, Anthony, for the introduction, and also to the foundation and to the program chairs, Louis and Jennifer, for the honor of speaking here today. My title is an overview of the clinical data on immunotherapy for cholangiocarcinoma, and I'd like to focus one, on the fact that this is a very clinically oriented talk, and I'm also want to make sure it's very clear from the outset that this is a work in progress, that the data I'm going to be presenting are largely trials that are very early, not yet published, perhaps just recently presented in most cases. And so I think I'll probably leave you at the end of this with more questions than answers, but the charge here, and I think it's an important one, is to provide an update of the data as it stands, since there have been so many new developments and changes along the way since last year's meeting. So here's that outline for today's talk. First, I'll start with a brief rationale for immunotherapy in cholangiocarcinoma, which fortunately has been covered beautifully by Sumera, and I'll just cover a couple high points from the clinician perspective. Next, the safety and efficacy of immunotherapy in cholangiocarcinoma clinical trials to date, including checkpoint inhibitors as monotherapy in various combination approaches that have been tried so far, and briefly mention a few of the novel strategies on the horizon. I'll then talk a little bit about something that's incredibly important as we approach immunotherapy in cholangiocarcinoma, the fact that this is not a one-size-fits-all cancer, and we can't expect any one strategy to work for all tumors in such a heterogeneous disease. And so that brings us to the important topic of biomarkers. And then, of course, we'll conclude with what are our next steps and future directions. So again, talking about the rationale for immunotherapy responsiveness in cholangiocarcinoma, as I mentioned, cholangiocarcinoma is extremely heterogeneous, but we know that there are subgroups with carcinogen exposure or other causes of underlying inflammation, such as fluke infection or viral infection, viral hepatitis, or non-alcoholic fatty liver disease, which are characteristics which are associated with immunotherapy responsiveness in other tumor types. So cholangiocarcinoma also has subgroups with higher mutational loads, though overall the tumor mutation burden is low. We see from the top signature 1 slide there that there are clusters defined by Jusical et al. Based on genetic, epigenetic, and gene expression profiling, and cluster 1 has a much higher gene mutation burden than the other 2 clusters, again highlighting the heterogeneity and the fact that some types of cholangiocarcinoma might be more predisposed to immunotherapy response based on certain features like tumor mutation load. And similarly, we know that one of the targets of immunotherapies, checkpoint inhibitors in particular, the programmed cell death protein 1, or PD-1, and its cognate ligand PD-L1, as Sumera has already talked about so well, can be upregulated in intrahepatic cholangiocarcinoma in particular compared to non-tumor liver, and in particular subsets within cholangiocarcinoma. So turning to the clinical trial data we have, I'll start with the monotherapy data, then some of the combination approaches. So the first slide here shows a host of early phase checkpoint inhibitor monotherapy studies that we have to work with so far, from Pembrolizumab and the KEYNOTE-28 trial that was first presented in 2015, not yet published, to Nivolumab, presented by Richard Kim and colleagues from Moffitt at ESMO GI in 2018, as well as Nivolumab in a Japanese cohort from Dr. Ikeda et al. And Durvalumab from Dr. Yoka et al., both presented at GI ASCO just a few weeks ago. So again, with the caution of the hazards of cross-study comparisons, and particularly these small phase 1B or phase 2 studies ranging from 20 to 40 patients, there are some, I think, important take-home points or signals we can draw. First, with the question of, is immunotherapy safe in cholangiocarcinoma, a tumor predisposed to inflammation and complications of biliary obstruction? Reassuringly, we see the treatment-related, or TRAE, rate is very similar to what we expect in other solid tumor types, relatively low. And while I don't show it here, the rates of hepatic toxicity are also reassuringly low, and we don't see any signal of increased hepatic inflammation. We also see some discrepancy in response rates from these monotherapy studies, where some studies report up to 17 to 19% response rate, others much lower, 3% and 5%. And that's where we come into the challenge of interpreting such small single-arm studies, where we realize there's quite a bit of heterogeneity in these trials, both anatomically, where they have different proportions with extrahepatic, intrahepatic, and gallbladder, which certainly could impact response. And also, there are some important biomarkers, which we'll come to, that are not clearly defined in these cohorts to date, particularly the presence of mismatch repair protein deficiency or microsatellite instability. Now, I'll return to that point in a moment. But first come to the largest study of checkpoint inhibitor monotherapy to date, which is the KEYNOTE-158 biliary cancer cohort. Now, KEYNOTE-158 was a basket trial of, I believe, over 20 different tumor types and enrolled 104 patients with biliary cancers. The majority were cholangiocarcinoma in a biliary cohort that was presented at ESMO 2018 in October. And so this trial is helpful for us to place the earlier monotherapy cohorts into context, in that it was a much larger study with centrally reviewed radiology, as well as patients were all tested for the important biomarker of microsatellite instability, and those patients were excluded from this cohort. So what we can see, again, returning to what can we interpret from a single-arm study here, the immune-related adverse events in this study were quite low, reassuringly only about 5% of the grade 3 or 4 level of toxicity, so a very well-tolerated treatment overall in this cohort. But what we see in the response rate category is only about 5.8% or about 6% of patients had a confirmed response on central review. And while it was a little bit higher by PD-L1 status, tumor PD-L1 expression, again, we'll talk about biomarkers in a moment, the numbers were too small to determine if that was significant. What we can say about those responses, though, is that like other immunotherapy responses in other tumor types, they were extremely durable, with the median duration of response not reached. So though the number was low, the quality was robust and meaningful. So I've mentioned already a couple times mismatch repair protein and microsatellite instability is a biomarker. And so just to cover that topic more clearly, mismatch repair deficiency refers to mutations in DNA mismatch repair genes, which are responsible for maintaining the integrity of our genome as the cells divide. There can be mutations in those genes, however, that can either happen as in adulthood over time or hereditary, that we can inherit from our parents. In that case, it's called hereditary non-polyposis colorectal cancer, GM-CSF, also known as Lynch syndrome. In cholangiocarcinoma, the incidence of this is quite low. This particular molecular aberration is about 2 to 3%, as Dr. Goyal already mentioned. But when it happens, just like in other solid tumor types, patients experience high rates of mutation in recurring stretches of DNA called microsatellites, which are present throughout our genome and refer to a PCR-based detection of microsatellite instability, so the genetic scar of the mismatch repair protein deficiency. And this can lead to a phenomenon called frame shift mutagenesis, where those little mutations throughout the genome can sometimes make a protein that looks aberrant to our own T cells and can be recognizable to the immune system. So when we look at this cohort of patients with mismatch repair deficient or microsatellite high solid tumors treated with checkpoint immunotherapies, there are several cohorts that are worth note. There's the KEYNOTE-16 basket trial is about 86 patients with deficient mismatch repair tumors. A couple of those 4 had cholangiocarcinoma. The overall response rate was 53%, including 21% of patients with complete responses. And those were very durable. The median also was not reached. And similarly, in other basket trials, prolonged responses and a high response rate of over 30% was seen, including subgroups with cholangiocarcinoma. So based on the strength of this signal, the US FDA approved Pembrolizumab for deficient mismatch repair or microsatellite high solid tumors, agnostic of the type of tumor. So regardless of whether it started in the liver or the colon or the breast, it is approved because of the strength of these responses since 2017. So I think the take-home message from this is that mismatch repair protein testing is warranted in advanced cholangiocarcinoma on the chance of that 2 to 3% incidence. So here's a slide that I showed just to exemplify this with a real case. I've shown this a couple times at this meeting over the years, and this is one of my patients who had a complete response to the PD-1 inhibitor Pembrolizumab on the KEYNOTE-28 trial. after progression on standard chemotherapy. We found after the fact that she had a tumor that was sporadically microsatellite high, so not inherited, but just something that occurred in her tumor. And she had a complete response, which endured for 2 years on treatment. Then per protocol, she stopped treatment. And as of now, as of just last week, she remains disease-free with no sign of cancer, 2.5 years off drug and almost 5 years since she originally started. So again, the strength of this response is worth noting and is what we are aiming for with these drugs. And it's also, and when we think about the clinical trials we're interpreting in some of the slides we've just shown and the ones I'm about to show further, it's an example of how a strong predictive biomarker can impact the results of small single-arm trials if we're not aware of its presence or not testing for it and making sure it's balanced. So to summarize, checkpoint inhibitors is monotherapy in cholangiocarcinoma to date. These are well tolerated so far. The treatment-related adverse effects seem similar to what we see in other solid tumor types. But the low overall response rates are not as high as we expect in other tumors or as we hope for. I think the benchmark is Keynote 158 with its response rate of about 6% by central review. These responses are durable and meaningful when they happen, but just a lower frequency than we need. And we do not know yet which anatomic or genotype subgroups are most likely to respond. So turning to checkpoint inhibitor combination strategies, a way to potentially improve upon this response rate by combining with another strategy. So this diagram wasn't specific for cholangiocarcinoma, but I think highlights the potential approaches one can take. And this is, we're drawing from lessons learned in other solid tumors. One approach is to combine checkpoint inhibitors with other chemotherapy drugs to potentially increase exposure to other tumor antigens and boost the immune response. Potentially combining checkpoint inhibitors with other checkpoint inhibitors, so PD-1, PD-L1 with CTLA-4 or other immune priming approaches to help boost the immune response. There are also studies of checkpoint inhibitors plus targeted therapies. I'll show a couple examples of therapies targeting other signaling pathways that have anti-cancer effects, such as TGF-beta or the MEK inhibitor, cobimetinib, or anti-angiogenic drugs like ramucirumab have all been studied in other tumors, and we have some early data in cholangio. And finally, there's an approach of great interest, but with very limited data so far, of combining immunotherapy with a liver-directed therapy, or such as radiation or ablation, to try to help release those tumor antigens and spur the immune response. So I'll quickly discuss a selection of these combinations that we have some early data from. One is with chemotherapy. Again, in lung cancer, we know that platinum-based combinations with chemo may do better when combined with checkpoint inhibition, though it is context and schedule dependent. There are several such trials underway in cholangiocarcinoma with GEMCIS, FOLFOX, CAPOX, taxane, plus various checkpoint inhibitors. The first to be reported, I believe, was GI ASCO, or the first that I'm aware of was GI ASCO just a couple of weeks ago, of Nivolumab in combination with gemcitabine plus cisplatin in chemotherapy-naive patients. The toxicity, it's noteworthy, is higher in combination than with a checkpoint inhibitor by itself, as one would expect, but the majority of these side effects are the standard chemo side effects, so not unique to the checkpoint inhibitor. The response rate was much higher, 37% on central review in this study, and the median overall survival is provocative, 15.4 months in a first-line study. So while these data are quite encouraging, particularly the overall response rate and overall survival, right now we can't tell from a single-arm study what is the component of contribution from the chemotherapy versus the Nivolumab, noting in particular that with a heterogeneous population with intrahepatic, extrahepatic, and gallbladder, we might actually see response rates that high with GEMCIS alone, and we don't know the duration of these responses yet. Are they a real immunotherapy response lasting over a year, or are they a couple of months like just a chemo response? So that's the data we need, and randomization is the key when follow-up studies are planned. Checkpoint inhibitor combinations have also been studied extensively in other tumor types, and we have now some data of a combination of durvalumab, which is a PD-L1 inhibitor, plus another checkpoint inhibitor called tremelimumab, in a Japanese or Asian cohort. And the take-home message, I think, from this very small study, or actually medium-sized single-arm study, 65 patients, was that it was also a tolerable combination. We're not seeing a safety signal. The response rate was a little higher, perhaps, than monotherapy of about 11%, but certainly not a home run for all patients. And the patients who respond still remain a cult to us. An interesting combination that's relatively novel in the scene is a new way of combining a PD-1 inhibitor, a checkpoint inhibitor, with another targeted therapy with TGF-beta trap, a bispecific inhibitor called a drug called M7824, which is a single antibody that can both block and inhibit TGF-beta function, which promotes the EMT as well as metastasis in various ways, as well as activating T cells through PD-1 inhibition at the same time. So a very provocative concept, and in fact, a phase 1 trial in 30 Asian biliary cancer patients showed an overall response rate of about 20% by central review, potentially higher in the small intrahepatic subgroup, and certainly intriguing signal warranting further study, and I'm certainly looking forward to seeing the next steps with this drug. We have a trial from UCSF where we've tried combining the checkpoint inhibitor Pembrolizumab with a cytokine growth factor called GM-CSF in hopes that doing the combination may also help prompt the immune response more than Pembrolizumab alone. And this is based on data from our colleague Larry Fong in prostate cancer that shows that GM-CSF can help recruit macrophages and other immune cells, particularly dendritic cells and T cells, to the prostate tumor microenvironment, and we're testing that in cholangiocarcinoma patients as well. And that study is currently in an expansion cohort with ongoing PBMC and paired biopsy collections to try to understand if we are achieving that in the immune microenvironment. And we've been encouraged to see a subset, about 19% of patients with prolonged responses so far. So in the interest of time, I'll switch to before concluding, to a trial that's ongoing but worthy of note as the first randomized trial in immunotherapy for cholangiocarcinoma, which is a checkpoint inhibitor atezolizumab plus the MEK inhibitor cobimetinib, based on the premise that if you inhibit with a targeted drug like cobimetinib, the MEK pathway, there can be favorable responses on the immune microenvironment to promote immune response. And so in this case, the trial design is taking atezolizumab as the control arm, a checkpoint inhibitor, compared to the targeted MEK inhibitor cobimetinib plus atezolizumab. And that study recently completed accrual through the Early Therapeutics Clinical Trial Network sponsored by the NCI, and we can hope for results sometime in the next year. So there are many other novel approaches in development, which again I'll skip over in the interest of time. We don't have any data yet to specific cholangiocarcinoma cohorts. So to summarize checkpoint inhibitor combinations in cholangiocarcinoma, the safety appears expected and similar across studies to all solid tumors. The response rate still is lower than we'd like, in the 20% or less for non-chemotherapy-based combinations, but the responses can be durable and meaningful. We need to determine the combination, the biomarkers of response and subgroup analyses to really understand which patients are likely to respond. And I'm going to skip to the question here of one of the questions I think that bears answering right now is, do driver mutations or tumor genotype impact checkpoint inhibitor response in cholangiocarcinoma? We heard from Dr. Goyal earlier about the increasing awareness of particular mutation pathways that drive oncogenesis in these cancers that are susceptible to targeted therapy. When we compare to other tumors with similar stories, such as lung cancer with ALK fusions or EGFR mutations, it's interesting that those driver populations seem to have lower response rates to immunotherapy, perhaps because of a lower tumor mutation burden. And while that's still an early emerging story in lung cancer, the data is recurring across cohorts, and I think bears us studying in cholangiocarcinoma as well to find out if checkpoint inhibitor as a monotherapy or even a combination strategy is the right approach in these driver mutations. So to conclude, checkpoint inhibitors can show a consistent high overall response rate in microsatellite high and mismatch repair deficient tumors, and certainly warrant testing for this phenotype in cholangiocarcinoma patients or for this mutation, MSI or MMR status, either one. Checkpoint monotherapy has a disappointing response rate, but those responses are durable and something we want to try to learn how to harness, potentially through combinations and also through identifying biomarkers to identify those patients who can respond. We need randomized and stratified trials to discern benefit from these combination studies, such as the NCI 10-139 trial of cobimetinib plus atezolizumab versus atezolizumab, and of course a continued effort on preclinical models and translational research to identify biomarkers. And I think to answer the call to collaboration, I think an early step we could take, particularly from these monotherapy cohorts, is to generate pooled sub-analyses of these initial checkpoint inhibitor trials to try to look at response rates and other efficacy parameters according to anatomic site, tumor genotype, bearing in mind the driver mutation considerations I just mentioned, and also PD-L1 status, and I think we would have a meaningful number of patients if we do that. And though challenging, immunotherapy can achieve meaningful responses in cholangiocarcinoma, and it has never been an easy task to study this heterogeneous disease, but we need to continue focused efforts on understanding how to do this. And this is another model that I've drawn from, I think, a lung cancer paper describing different strategies for how to approach different immune biomarker cohorts once we start to understand them and try to eventually reach a day that we can find the right combination approach for the right patient. And I will conclude. Thank you.